Guys, after subscribing to this channel, please make sure that you also press the bell icon so that no notification, no new video of mine, any educational video, is missed by anyone.
One question, as we've all talked about genetic analysis on the pregnancy tissues. Can somebody tell me what analysis do we want? What tests would you write down? You want genetic analysis of pregnancy tissue. I want to know which analysis do you want? Who sent me these beautiful charts, these tables? Who's the one? Ashwini, is that you? Usha, is that you? It's in between both of you. So who has sent me that? Who sent me those tables? Usha, did you? Oh, beautiful table, beautiful table. Yeah, it's very much like my own table. Thank you, Usha. You are a darling. Really, I'm so impressed with this girl. Her dedication level is too good. She's there in all of my classes, a very, very active participant, and I want all of you, to imbibe this energy. This is what you get in the live classes, which you will never get in a recorded version. Okay, so try to attend my classes on time and be there. It's not a very big deal to take out 1 hour from your daily schedule. Okay, I'm going through a lot of rigorous training, structural programs right now, but I still am able to not only make the class, also present it to you in 1.5 hours. Just promise me to be around for this class. You'll imbibe the energy and you will understand how good the other people are doing, and then you'll also get that positive vibe to do yourself. Okay, that's my idea, to infuse a lot of energy into you, a lot of positive vibes inside of you. Somebody is doing so good, you get inspired. Okay, and I want you to take home that energy as well. Take home message is fine. Take home sentences and slides are fine, but take home energy is another level. Okay, yeah. So please tell me, what all do you think? Okay, CMS 1, very good. Chromosomal microarray, most of you know about that. Karyotyping is another one. What else? Has anybody spoken or thought about next generation sequencing, exome sequencing, whole exome sequencing? Has anybody spoken about it? Okay, Ashwini has written down whole exome sequencing, WES. Yes, correct. Absolutely. CMA is very important. So let's just discuss about these as well. Small. Conventional karyotyping. You know what conventional karyotyping is? All those people who were there in my class, I used to talk to them like this. For example, if something is wrong with the car, something goes wrong with the car. And what you're seeing right now, what is karyotyping, is that you're seeing the car from outside. Okay. So you can see that the paint is lost, or there is a dent in the car, or the wipers are not working fine. But would you be able to say what has happened to the carburetor or the radiator or the fuel? No, because for that you'll have to open it up. So karyotyping is like seeing the car from outside, right? So it's limited by culture failure, pooled rates of approximately. So what you do is you culture them and then you spread it out, and then you see by structure. So basically you're seeing the structure of the chromosome. If you have aneuploidies, if you have trisomies, monosomies, translocations or deletions, and that are big deletions, deletions, duplications. So that's basically karyotyping for you. Then you have something smaller, more refined than this, is FISH. Okay. So these are smaller, the micro deletions, micro duplications, they'll be able to get through FISH. So you're taking these fluorescent tubes, this bind to specific parts of chromosome. Okay, so you have certain specific probes which are utilized, like for example, chromosome 13, 16, 18, 20. Usually you have problems with these chromosomes. Okay, and again, here you have culture failures, correct? And then you have comparative genomic hybridization. Okay, chromosomal microanalysis is, array CGH is actually a form of CMR. I'm sorry, CMA, chromosomal microanalysis, CGH. Comparative genomic hybridization is a form of chromosomal microanalysis based on the use of differentially labeled tests and reference genomic DNA samples that are simultaneously hybridized to DNA targets, arrayed on a glass slide or other solid platform. Now, it avoids the limitations of conventional karyotype and FISH, just culture failure, limited chromosomal examination, and essential scans the entire genome, providing additional information such as DNA copy number variancy. All these smaller parts, now you're able to go and see what is exactly wrong with the engine, or with the radiator, or what is wrong with the fuel supply, or somewhere where the oil is leaking because you've gotten inside the car. Okay, so these are finer tests. Okay, And then single nucleotide polymorphism is another level. Okay, now you're seeing, you've opened up the engine and now maybe you're seeing a part of the engine. So this is what it is. It is even finer. This again it's a kind of chromosomal microarray. So if anybody comes to tell you that you do a microarray of POCs, you'll ask them which one. Microarray, chromosomal microarray, which one? Is it CGH? Is it SNP? What? So here you can additionally detect the parental source of chromosomes and anomalies and can therefore be used to rule out any maternal cell contamination. So this is a one step further in understanding the products of conception. What is the problem? This is another, platform. Okay. So NGS is next generation sequencing. It is at the level of nucleotide resolution. Do you know how DNA is made? By nucleotides and nucleosides. Okay, so it goes to that level. It sees whether the sequencing is right or not. So nucleotide polymorphism, it was seeing the whole, that sequence. CGH, CGT or TGU, all these things, right? But this is seeing C alone, G alone. This is what it is. Nucleotide resolution. It scrutinizes the entire genome through tiny quantity of tissue. So this is at the very, very micro level. Expensive, currently expensive, and requires data processing because it's a very large data output. So every nucleotide is being scanned. So it's at another level. Okay, this next generation sequencing, but nevertheless, these names. You have next generation sequencing, single nucleotide, and SNP and CGH. They are both forms of microarrays. So next time, if somebody asks you or tells you all these, these genetic lab tests, they come to your clinic and they tell you that send the POCs for microarray. Ask them which microarray you're talking about. You're talking about the expensive SNP or you're talking about the CGH, because both are doing well. Okay, CGH, if you really have a requirement of SNP, then only go for SNP. Okay, just give me 2 minutes. All right. So guys, I think with this, we have finished the most important aspect of the recurrent pregnancy losses. Now we know the risk factors, we know the investigations, and we know their management. Now, if you have any questions, I'm open to questions, and we still have 10 minutes to talk. So let the questions be. And yeah, I want one volunteer for my next class who's going to take up the case of recurrent pregnancy loss. And one more thing, guys, you can easily make the the cases. Okay, we used to do that very often because when I was studying for DNB, at that time we were not going to the clinic. Okay, we were not going to the institute. So we went in for, I went in for certain fake cases as well, which were made all by my thought process. And question of Ashwini is, what is the management of normal thyroid and positive TPO with recurrent miscarriage? You don't do anything, guys. Yeah, you keep repeating her thyroid test, because at some point in time these anti-TPO antibodies are going to cause hypothyroidism. Then you can supplement. Otherwise, you will not supplement. You will just follow her up, because these anti-TPO antibodies, they're not strong enough. That means that they are causing any clinical or subclinical hypothyroidism, and they are not per se the reasons. Again, over here, if you're looking forward to American association as well, even they have not said supplement thyroxine. They have not. So you will do nothing. You will just follow her up. Okay, and you'll see if she gets pregnant, and then you follow her up at 7 to 9 weeks again. Okay, so this you want me to include, Usha? Criteria for APS? All right, so I'll do that as well. If she wants to conceive, then she can go ahead and conceive. You can just tell her that there's not even subclinical hypothyroidism, and it's just the presence of anti-TPO antibodies, and you don't have to do anything for that. Autoimmune reason, that too anti-TPO antibodies alone with normal thyroid is not a recommendation for supplementation. Please go ahead and get pregnant. Try to get pregnant. Okay, yeah, that's the recommendation. You are not doing anything for this patient. You're just following her up. Just tell her that they are not strong enough to cause, and per se alone TPO antibodies, they are not associated enough to be the reason for your recurrent pregnancy loss. And if she still asks you, then what is the reason? Well, if now it is unexplained, we can't say. but we'll give a very strict watch on your level of thyroids, because the moment they start deranging your thyroid levels is when we supplement you with the thyroid. Otherwise, we are unnecessarily prolonging your treatment, which we actually don't need. Okay, I'll add this, Usha, for sure. Anything else which you feel that it should be added, please be honest. I have no problem at all. It's not, see, this was actually GTG, so no problem in that. But I remember that this criteria was not even, ULAR criteria was not there in my last class as well. So I'll put in, no problem. Anything else, guys? You want to ask? You want to let me know? Okay. So I'll wind up the class, and in that case, you are more than welcome to me at any time with your queries. All those students who keep asking me questions will vouch that I answer their queries as soon as I see them. So there is no point in of delay. If there are certain things which I need to maybe check on and then get back to you, I always ask my students to remind me, because sometimes so happens that I get, if I've answered them immediately, or I tend to forget. So please remind me in that case, okay? Thank you so much for all of you for attending this class, as usual, for making it so enthusiastic, so alive, and so interesting for me, okay, and for everyone else. Thank you for your positive energy, everyone. And I see you around next Saturday, hopefully, at the regular time. Okay, thank you. Yeah, it'll be on Saturday at the regular time, okay? I was busy last Saturday. That's why I could not take it. But now it's going to be on Saturday, okay? All right, guys. Thank you. God bless everyone. Enjoy your Sunday.